Hello class, today we are going to be learning about meiosis. Can anyone tell me what it is?
Meiosis is a reduction division of a diploid nucleus to form haploid gametes.
Very good. Body cells or somatic cells are diploid, they contain a homologous pair of each chromosome. Does anyone know what a homologous chromosome is?
Homologous chromosomes are chromosomes with the same genes as each other, in the same sequence but do not necessarily have the same alleles of those genes.
Great. Now everyone turn your attention to the board for a short clip outlining the process of meiosis.
Meiosis has two main phases. Meiosis 1 is reduction division, which separates the chromosomes of homologous pairs. Meiosis occurs in eukaryotic cells. The first stage is prophase 1. The nuclear membrane begins to break down and the DNA begins to supercoil. Through a process called synapsis, homologous pairs of chromosomes are brought together. Crossover begins in prophase and finishes in the next phase, which is metaphase. This is the exchange of genetic material between non-sister chromatids. The non-sister chromatids cross over at points called chiasmas. This creates a genetic diversity of gametes. The next stage is metaphase. In metaphase, the homologous pairs line up on the equatorial plate in a random fashion, thus creating genetic diversity. They are in their most supercoiled and condensed state. Spindle fibers attach the chromosomes to alternative poles. Crossing over finishes here. In anaphase 1, the spindle fibers or microtubules contract and pull the homologous pairs and their alleles away from each other. The sister chromatids move to opposite poles. In telophase 1, each pole of the cell contains one of the sister chromatids of the homologous pair. A cleavage furrow forms and the cell splits into two. Meiosis 2 splits apart the sister chromatids. The first stage is prophase 2. The DNA condenses again and the sister chromatids begin to move toward the equatorial plate. Microtubules readjust to specific positions in order to split the DNA properly and attach to the centromeres of the sister chromatids. In metaphase 2, the sister chromatids are aligned on the equatorial plate and ready to be split apart. Anaphase 2 is where the spindle fibers contract and the centromere motors pull on the microtubules so that the sister chromatids pull apart. A chromatid from each pair goes to each pole. The chromatids can now be referred to as chromosomes again. The chromosomes from the pair of sister chromatids are not identical due to the exchange of DNA during crossover. Finally, in telophase 2, the chromosomes gather at opposite poles to form the new haploid nuclei. The nuclear envelope material that has remained within the cytoplasm reforms around the sets of chromosomes. There are now 2 nuclei per cell, each is haploid.
Does anyone have any questions on meiosis?
What happens if, something goes wrong during meiosis and the chromatids of those sister chromatid thingies split incorrectly?
This is called a non-disjunction, an error in meiosis that produces cells with unusual combinations of chromosomes. An example of this is Down syndrome. This occurs when the homologous chromosomes fail to separate during anaphase 1 of meiosis, or Y chromatids fail to separate during anaphase 2.
Then the daughter haploids would have either 0 or 2 chromatids. So when these gametes fuse with a normal haploid gamete, the zygote will have an odd number of chromosomes, right?
Correct. Down syndrome occurs from a non-disjunction in the form of a trisomy on chromosome 21. Does anyone know what a trisomy is?
Is it the presence of 3 copies of a chromosome?
Exactly. I do believe it's time for your break. Please be back in class in 15 minutes. Thanks.
So I was thinking, my siblings and I are so different. Why is this?
This is because meiosis results in an effectively infinite genetic variety in gametes through crossing over in prophase 1, when genes are swapped between the homologous pairs.
Could it also be that the homologous chromosomes orientate randomly during metaphase?
That's right. The homologous chromosomes can align in a manner of ways, and different sister chromatids can be pulled to each pole. This links to Mendel's law.
What is that?
This law states that when gametes are formed, the separation of one pair of alleles between daughter cells is independent of the separation of another pair of gametes.
What does that mean?
This means that traits are transmitted to offspring independently of one another. Any one pair of characteristics may combine with either one of another pair.